Good day. In this part of the lecture, we will be discussing about molecular phylogeny. So, brief outline. So, we will look at the theories, the neutral theory of evolution and positive and negative selection, which is the basis for doing this phylogenetic trees. And we will look at what are the types and what are the parts of phylogenetic trees. And finally, we will look at how do we actually create these phylogenetic trees. So, before we actually do with the neutral theory of evolution, let's look at the selection, positive and negative selection. So, if you remember in the original theory of evolution, there is natural selection, whereas as the evolution, the species must change, must adapt, and that the one that is well adapted becomes selected. That means they survive to continue on their genes to the next generation. So we can observe them basically from in the molecular level. We can observe them actually in the recent pandemic. We have seen them in how the genome, the structure, the when I say the structure, that is the structural proteins of the SARS-CoV-2 virus is changing based, and that is why we have these many different strains. They mutate. You have strains, and only the strains that are able to infect many people faster, the strains that can evade the immune system much better, are the ones that are actually being selected. In fact, I believe the vaccines, no, not exactly I believe, but rather the vaccines itself are exerting a selective pressure on these viruses, because only the virus that can evade the vaccine can, of course, have a chance to multiply and infect new individuals. But anyway, here is the positive and the negative selection. So again, if a certain, for example, you have this population, each of us has a slightly different variation in our genomes. And for example, if that particular variation gives you a selective advantage uh, in survival, therefore you have this selection, it's a positive selection. So if there is a certain environmental event that happens that allows that particular variation to be more adapted to that change in the environment, of course that person will survive and propagate his or her genes to the next generation. So you will have this positive selection towards that variation. On the other hand, the negative selection is where this variation might cause a disadvantage, and it becomes a selection against that particular variation. So it will become lost. That particular variation will be lost, because of course the person that has that variation will not survive to pass on their genes to the next generation. That's basically the idea with the selection, the positive and the negative selection. And you can actually see them in the molecular level with regards to your genes. Okay, and we have now the neutral theory of this molecular evolution. Now, because we have this positive and a negative selection that gives advantages or disadvantages, now the neutral theory, on the other hand, posits that the most of the variations, the molecular level that we currently have, doesn't have much of an effect in the fitness. Therefore, the evolutionary fate of genetic variation is explained by stochastic processes. This theory, because again, it's not always the case that there's always a positive and a negative selection. Sometimes it's neither positive nor negative. That's actually where the neutral theory comes from. And this is because it's neutral, you can have this stochastic explanation or stochastic models to explain them. So you have Moto Kimura proposing a model to explain evolution at the DNA level. Based on the rate of amino acid substitution average, approximately one change per 28 times 10 to the 6 years for proteins of 100 residues. So in this model by Moto Kimura, it provides you with a timeline on how fast a certain amino acid substitution occurs in certain genome of a certain lineage. So it now gives, based on this model, it provides us with a data on how many years must occur for that. So again, this is still only a model. and not an actual fact. Now, Kimura's conclusion was that most observed DNA substitution must be neutral or nearly neutral, and that is the main cause of evolutionary change at the molecular level is random drift of your mutant alleles. So most non-synonymous mutations are deleterious and are therefore not observed as substitutions in populations. Now, again, the neutral, this focuses only on changes that gives neither a positive impact or that gives neither advantage nor disadvantage to the adaptation. It's just mainly that observed changes in the proteins that are neither helpful nor detrimental. And it provides you with a date. And that is why if you look at most evolutionary trees, some of the trees have dates. And that is where, because that is where those dates come from the model of Moto-Kimura. Now let's look at these trees. So a phylogenetic tree is one that estimates the historical connections between species or genes that carry. So when we say historical connection, it has this particular sense of a timeline on when this particular change occurs, this particular change that happened that gives you these changes in this amino acid based again on the model of the neutral theory. Now let's look at the parts of the trees. So here on this image an example of a phylogenetic tree. At the tip of the tree is the branch that represents that particular taxa of the study. So it can be a specific species, it can be an order, a population, etc. So, these taxa, we call them OTUS or operational taxonomic units. Now, the lines in the tree are the branches. The points which connect, the branches connect, or the tips of the branches are the nodes. So, the nodes are the branching point of a certain branch. So, that node is where that is actually a hypothetical organism. It's not this is actually one of the controversies and conflicts about these trees because the nodes represent hypothetical species that are not actually real, and that is where the problem is, or the controversy is with the use of these phylogenetic trees. Now, aside from that, we have the root, the basal node or the root for the trees. Now, we usually, when we do phylogenetic trees, we sometimes use as a root or a basis of the root as a certain animal that is very vastly different from the group that you are comparing. For example, you want to compare the different vertebrates, so the cat, the dog, the mouse, etc., etc., and then you add to make your tree rooted, you add a certain animal that is vastly different. So you add, for example, an insect. For example, here, comparing human and mouse, and then you add a fly, which is vastly different from that group, to give us an idea of the root. So grouping an ancestor and all of its descendants is known as a clade. Basically, this image can also be considered a cladogram. Okay, so what even amidst all of the controversies of this phylogenetic tree, why do we still have this? Why do we still have these phylogenetic trees? So the goal basically of this analysis is to recover these bifurcating trees in which each taxon is linked to one other taxon through a node. This is basically how the current way to group animal species, animal orders, animal classes with one another. So it gives us an idea of whether, for example, as humans, are we closely related, which are we more closely related to, a mouse or a rat? Basically something like that. Or is it, are we as closely related to a mouse, or are we as closely related to a horse? Which is closer to us? That is the goal of that. And why do we want to know which of these species are more closely related? It has something to do with the use of animal models, especially